Hey everybody, I hope you are enjoying your weekend wherever you are. Today is a special day. It's February 29th, leap day, because it's a leap year. So, and we had lecture on Thursday, and today I'm going to review what we went over on Thursday. Sorry. So today we are going to start talking about transcription, and before we get into transcription, we are going to talk about the genetic code. Now, before we get into that, let's review some important terms. We have transcription, which is the process of how DNA is used to make RNA, and we have translation, which is the process of how RNA is read or translated to make proteins. And in our last lecture, we saw a case of how RNA was used, or the code in RNA was used to synthesize DNA, and that was called reverse transcription. And if you don't remember, where did we see this? We saw this with telomerase. So telomerase used in RNA primer, well, not RNA primer, an RNA template. That was the Turk molecule. So we'll do a little review here, which served as the template to lengthen the telomeres. So that was an RNA molecule that the enzyme used to extend the telomeres. Okay, so with that, we need to talk about one more thing. We're going to talk more about proteins, I believe, in next week. So with respect to proteins, for now, let's just remember that they are chains of amino acids. And the order of the amino acids in the chains is determined by the order of codons in a messenger RNA. And a codon is going to be 3 nucleotides long. And the order of the codons in a messenger RNA determines the order of amino acids in a protein. And the main part or main theme of this video right here will be on the genetic code. Specifically, how was it elucidated? Elucidation. How was each codon in the genetic code matched up with the amino acid that codon specifies? So we're going to talk about this story right here. It's kind of neat, and we get to learn a lot about the experiments that were used, and I think it's really interesting. So let's take a look at this process, or the story of how the genetic code was elucidated. And we won't be able to talk about all the experiments were done. We will introduce the initial experiments, and then we'll have to skip over a lot of the other experiments that were performed and get to, and we'll end with the most important experiment that really helped scientists wrap up the genetic code. So the story starts with some nucleoside diphosphates. So ribonucleoside diphosphates. And so a little review here. Remember, nucleotides are molecules containing the 5-carbon sugar, a nitrogenous base, that's going to be adenine, cytosine, guanine, thymine, or uracil, and a phosphate group. Now, a nucleoside is just the 5-carbon sugar and the nitrogenous base. Nucleoside. So add the nucleoside to the phosphate. Combine a nucleoside with a phosphate group and you have a nucleotide. So here, a ribonucleoside diphosphates, that's just a neat way of indicating a ribonucleotide with 2 phosphate groups. And why are we saying ribonucleotide? So in this course, we may say nucleotide. We may need to specify ribonucleotide, or we may need to specify deoxyribonucleotide. So the general term here, nucleotide, refers to both possibilities, and normally you can determine which one we mean when we say nucleotide by the context of the discussion. And sometimes we need to specify because it might not be so clear which one we mean. So ribonucleotide, these are the nucleotides in RNA. Deoxyribonucleotides, these are the nucleotides in DNA. And maybe you remember from our discussion of nucleotides, I think back in lecture 10, that the key difference here, other than uracil being in RNA and not DNA, and thymine being in DNA, the main difference is the 5-carbon sugar. So ribonucleotides have ribose as the 5-carbon sugar, and deoxyribonucleotides have deoxyribose. And why don't you take a second to think about the structures of those? We needed to know the structures of these, right? Remember these? So they're very similar. So here's the structure with 1 carbon here, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So these are all the carbons. There's an OH here. The oxygen has to be bound to this 3 prime carbon, not the hydrogen. We bind the oxygen here. Now, the main difference between ribose and deoxyribose is what's here. So when we don't put anything there, essentially that means there's a hydrogen there. We just don't typically put an H. And when there's an H there, that's deoxyribose. If there is an OH here, O is bound to the carbon, the H is bound to the O, that is ribose. So ribose has an OH group bound to the 2' position of the 5-carbon sugar. Deoxyribose just has an H. Okay, so back to the ribonucleoside diphosphates. These are going to be molecules that we typically put into an RNA chain. The diphosphate, what is that? So remember when DNA polymerase is synthesizing or polymerizing DNA, it is using deoxyribonucleoside triphosphates, right? It's using D, and I can put an N here, TPs. So deoxyribo for the D, nucleotide triphosphates. And this would be bound to the 5' carbon of the nucleotide. Sugar and nucleotide. Now, that's when DNA polymerase is synthesizing DNA. 3 phosphate groups. In a ribonucleoside diphosphates, we're just going to have 2 phosphate groups. So diphosphate, 2 phosphate groups bound to the 5-carbon sugar. And let me diagram that for you. So switch pencils here. Ribonucleoside diphosphate. So 2 phosphate groups. I'm going to put the O's here. Remember that the double bounded O's are usually, the double bond needs to be a free oxygen, not bound to anything else. So there's the 5' carbon, 4'. Oop. That always happens when I grab a new pencil. Okay, here we have 1, 2, 3, 4, 5, our carbons, 1, 2, 3, 4, and 5'. This one connects to the base, and we could use any base in here, right? The base could be adenine, cytosine, guanine, thymine, nope, not thymine, tricked you there, and uracil, right? Because we're talking about ribonucleoside diphosphates. And thymine is in DNA, not RNA. So there will be an OH here, and again, because this is a ribonucleoside diphosphate, there will be an OH there. Okay, and we can symbolize these ribonucleoside diphosphates as NDPs, nucleoside diphosphates, ribonucleoside diphosphates. I guess we could put an R in front of there if we wanted to. No, why not? RNDPs. Excuse me, taking a sip of coffee here. It's early Saturday morning. Hopefully my gulping that coffee doesn't show up on the video. It's a little embarrassing. Okay, so now we know what ribonucleoside diphosphates are. We are experts, hopefully, in what those are. Now the next thing scientists needed was an enzyme that could join these together. And, you know, I need to look this up. I can't remember the details. If scientists had already discovered RNA polymerase, which is used by cells to transcribe RNA, but they didn't know how to use it possibly, or maybe they hadn't discovered it yet. I think they discovered it and just didn't know how to use it to make long chains of RNA. Now, but they did know about an enzyme, polynucleotide phosphorylase. Polynucleotide phosphorylase. I think we will abbreviate that PNP. Now what this enzyme can do is, if you give it in a test tube, put the enzyme in a test tube with high levels, unnaturally high levels of NDPs. Levels are NDPs. Levels that you wouldn't see in a cell of A, C, U, and G are NDPs. A meaning adenine, C cytosine, uracil, guanine. What it can do, you know what, I should draw the arrows like this. So high levels of RNDPs with this enzyme will join them together. 5', U, A, G, U, U. It doesn't matter. I'm just putting random nucleotides in here because it just joins them together randomly. So on and so forth. Now, with this, scientists can make RNA. They can make RNA. And if you read through the notes, you'll get an extra detail here. What this enzyme normally does in cells is it breaks down RNA chains into RNDPs. And maybe when you take biochemistry, you can learn that enzymes can work in both directions. A lot of enzymes can. They can synthesize things, or they can break them down depending on the levels of products and substrates. I don't know if every enzyme that makes something can do that catalyzes a reaction can do that, but this is one of them. So it normally breaks this down, long chains down, into individual RNDPs. But if you give it high levels that it normally doesn't encounter, high levels of RNTPs that it normally won't see in a cell, it will, you know, do the opposite. It will make long chains. So I think that's all I want to, oh, and another, okay, so this is just like, this is an RNA molecule, and the bonds that are formed between each of these are 3' to 5' phosphodiester linkages. So just like you would get 3' to 5' phosphodiester linkages, just like you would get in an RNA transcript made by RNA polymerase. So this is a fully functional RNA molecule. And if we could load a ribosome onto it, we haven't talked about ribosomes yet, but I'm sure you've seen those in introductory biology course. If we could add a ribosome to here, and give it amino acids, transfer RNAs, everything it needs to translate an RNA, ribosome would translate this, because this is a fully functional RNA molecule. It looks just like an RNA molecule that is transcribed by RNA polymerase, which we will talk about in the next video. Okay, so with the discovery of PNP, polynucleotide phosphorylase, scientists needed one more thing to be able to actually make a protein in a test tube. So they needed the protein production machinery of the cell. They needed ribosomes, they needed amino acids, and transfer RNAs. But you know, they didn't have to wait to discover all of those things. They figured out a pretty cool way that they could make proteins, and I'm going to go over that now. So PPCL is the abbreviation I'm going to use for the protein producing cell lysate. So what some pretty clever scientists figured out was that they could take E. coli cells, and they could take a bunch of these cells, they say millions of cells, and they could lyse them. So break open the cells and release the cytoplasm and all the organelles in the cytoplasm. Organelles, I don't know if I can use that word right. Like I'll spell out all the maybe I can, I forget. I forget the details on some of these terms. So lyse the cells, and the ribosomes and the transfer RNAs, the DNA, everything will be spilled out with the cell lysate. So they can collect the cell lysate, they can centrifuge it, and then in that centrifuge tube, what they'll have is a bunch of cell debris down here, essentially anything that isn't soluble, like cell walls. Essentially, I only know cell walls as the thing that's not soluble, but there's other stuff in there. And up here, we will have the supernatant or supernate. Similar words you can use, supernatant and supernate, interchangeably. I don't know if it was always that way, but today we can use them interchangeably. So in the supernate, what was there? Well, they don't know. I don't know that they knew of all these things that were in there, specifically ribosomes, amino acids, transfer RNAs, elongation factors, also, you know, other translation factors. You don't need to memorize that for this part, for this video. These details we'll cover later. But the point I'm trying to make, everything the cell needs to make proteins is found in here. So this is the protein producing cell lysate, the supernatant. So everything in here, all the components needed to make a protein are in here. It didn't matter if scientists that knew, they didn't need to check, okay, let's make sure that this is here, this is here, this is here. That the supernatant could make protein, because it had all the proteins, all the things that were necessary, which makes sense, right? You take a cell that can make proteins, you spill out everything, and you use that lysate and everything in there to make a protein. So, okay, so now they had a way, PPCL, we're abbreviating it, now they had a way to make proteins. They had a way to make RNA, and they had a way to make proteins. So, first experiment is they took PNP, the polynucleotide phosphorylase, and they took uracil RNDPs, right? So just uracil. They're not going to take all the different types, just using uracil here. And this allowed them, that always happens the first time I grab a sharpened pencil. I had a student tell me once that my pencil wouldn't break so much if I wasn't pressing down so hard. And he probably has a good point. So, okay, so PNP plus uracil RNDPs, what does that make? A long chain of RNA containing just U's. We're going to call this polyuracil. Okay, so, and if you see this polyuracil, how many different codons do we see in there? Here's another one down here. How many different codons are present? So if we look right here, just U's, right? So we have one codon that just has 3 uracil nucleotides. We look here, 3 U's, oh, that's 2, sorry. 3 U's here, 3 U's here. There's only one type of codon. Okay, so all the codons are U. So essentially, if they could combine this polyuracil with the PPCL, they can determine what codon, well, let me show you how it works. They take polyuracil, combine it with the PPCL in a test tube. And I'm going to show 3 different test tubes here. So, and there's the top of the liquid in the test tube. Here, PPCL has been added to each one. Poly-U has been added to each one. And every tube is going to get one thing different. They're each going to get a radioactive amino acid. In the isotope, the scientists are using our 14C. Now, what does this mean, essentially? Essentially, it means, I think carbon, man, I should have looked this up. Carbon normally has 6 protons and 6 neutrons. And that's carbon 12. Carbon 14, I hope that's right. Carbon 14, however, would have 6 protons and 8 neutrons. And if you're watching this video, you might want to just take a quick look at a table of the elements and make sure that, you know, carbon typically has 6 protons. So 6 protons and 8 neutrons. This is unstable. We're going to call this radioactive. And scientists could obtain amino acids that contain 14C instead of 12C or 13C. I believe 13C is a stable, somewhat stable form isotope of carbon. But they were using 14C, and it's unstable and radioactive. So they can detect amino acids with 14C in their structure. So for this tube right here, back to the experiment, let's say they added 14C phenylalanine. Phenylalanine, I'll abbreviate that Phe. So that's an amino acid. It's a radioactive amino acid. This tube has other amino acids in there from the cell lysate, but it only has one radioactive amino acid. That's 14C phenylalanine. To this one, let's say they added 14C tyrosine. And for this one over here, let's say they added 14C alanine. Okay, and they do this experiment, They added the polyuracil, they added PPCl, they added 14C phenylalanine, and they let the reaction incubate for a certain amount of time. Then they isolate proteins. So when they isolate the proteins, they don't get the free amino acids. They purify that protein that was synthesized in this test tube, and they free it away from any free amino acid, such as 14C phenylalanine. And when they look at the protein is radioactive. So the protein that was made in this test tube is radioactive. This one, no radioactive protein. No radioactive protein synthesized. And I realize I'm writing too small here. Hopefully you're listening and being able to interpret what I'm writing. So here, no radioactive protein synthesized. So they did this experiment 20 times, one for each amino acid. And what they found was that only one of the test tubes, only one of the reactions resulted in a protein, a radioactive protein, and that's this one right here, the one where they added the phenylalanine, the radioactive phenylalanine. So we can conclude that the codon 5' UUU, which is the only codon present in the poly-U RNA, codes for phenylalanine. Okay, so they were able to pretty much, conclusively, very convincingly show that this codon right here of 3 U's corresponds to the amino acid phenylalanine. Now that's pretty good, right? That's a neat experiment. And how many codons are there anyways? How many possible codons are there? So let's see. So if a codon is made up of 3 bases, or 3 nucleotides, 1, 2, 3. So there are 4 possible nucleotides at this position, 4 possible nucleotides at that position. Let's say this is the 5' end, this is the 3' end, and 4 possible nucleotides at this position, right? For each one, it can be A, C, G, or U. Each one of these can be A, C, G, or U. I could even put a little or here, or U, or U, or U in commas. But I think you get the point. So to figure out how many codons there are, we could simply just multiply this 4 by 4 by 4. And you don't really need to understand the mathematics behind why this works, but 4 by 4 by 4, 64 codons are possible with a 3 nucleotide, because codons have 3 nucleotides, and there are 4 types of nucleotides. So UUU is just 1. Right? UUU is just 1 codon. We need to figure out what the other 63 codons are now. So this was a huge finding, but okay, now they need to figure out what the other 63 are. And you could probably see that, okay, they could do the same experiment. PNP plus adenine are NDPs. Oh, I really butchered that one. RNDPs. Adenine ribonucleoside diphosphates. And what that will make. Take a second to predict what that will make. You know what? Go back to the other, back up the video, see what we did for uracil. Okay, what did that make? That makes long chains of, it joins long chains of A's together. So this makes a poly-A RNA. And when do the experiment with poly-A, what they found was, and what is the experiment? Adding poly-A to PPCl, protein-producing cell lysate, with each of the amino acids radioactively labeled 1 at a time, so 20 different reactions, 14C amino acid. What they found was that only the lysine, 14C lysine, gave a radioactive protein. resulted in a radioactive protein. None of the others worked. Okay, so what does that mean? That means AAA specifies lysine. So now we're down to 62 codons to solve. They did the same thing with PNP and cytosine, RNDPs. And what they got was, I won't diagram all that, I'll just tell you, they figured out CCC specifies proline. They tried the same thing, right? They tried the same thing for guanine, with guanine RNDPs. And unfortunately, that did not work. And it's kind of interesting. Guanine, long chains of guanine, tend to form what is called a G quadruplex. Quadruplex. Or maybe quadruplex. Again, my spelling's terrible. Sorry for my elementary school teachers. I probably wasn't paying attention in class. G quadruplex DNA. Not DNA. A G quadruplex structure. And what that means is essentially it's going to fold into some funky structure, which would reasonably make it hard to translate into to make a protein. So this one didn't work. For whatever reason, possibly because of the G quadruplex tendency of long chains of G to form. But we figured out 3, so 61 to go. I hope I'm not running out of space on my phone. We're at 34 minutes. Take a look at my notes here. Okay, so lots of other experiments were done over several years. I forget the timeline specifically. And they weren't very satisfactory because they were very difficult. And the problem was with PNP, right? So polynucleotide phosphorylase joins nucleotides. You know, I don't like this pencil. Let me sharpen this one. Typically I like these pencils, but it's not dark enough. It doesn't. Okay, the problem was with PNP or polynucleotide phosphorylase, and it was because of the way it joined nucleotides, right? It joins them randomly. So the scientists could never really be sure of what codons they had in a chain of RNA if they gave it more than one nucleotide. For example, if we gave it adenine, RNDPs, and cytosine, RNDPs, NDPs, it could make, something like this. And, we could kind of guess, maybe, okay, so all the codons would have to have A's and C's in them, right? A's and C's. But when we tried to use these to make proteins, and, we'd see some proteins or amino acids being incorporated into proteins more than others, but there was some level of guessing involved, and it was, they were able to solve some, but it wasn't very satisfactory. There was also a triplet binding assay, which was, I guess, a really interesting, a really useful experiment, and they were able to, I think, link up maybe a couple dozen of the codons with specific amino acids using this thing called the triplet binding assay. But because we're getting, you don't have all day or all semester to learn about the history of the genetic code, we're going to jump right to the end, to the experiments devised by a scientist named Gobind Khorana, who won the Nobel Prize for his work on deciphering the genetic code. So the PNP enzyme was great for figuring out the first couple of amino acids, or codons, figuring out the amino acids for the first few codons. But the random nature with which it joined nucleotides made it difficult to do much more with it. So scientists needed a different way to make long chains of RNA. And Gobay and Khorana came up with this neat assay called the repeating copolymer assay. So although scientists couldn't synthesize long chains of RNA with a specific sequence, what they could do is make very short chains of RNA of a specific sequence. For example, they could make dinucleotides containing any combination of two nucleotides. In this case, let's say U and C. So they could make millions of molecules of just U and C. And they could do the same thing with 3 nucleotides. So they can make trinucleotide molecules of any sequence they wanted. And let's say they made millions of nucleotides, of trinucleotides with U followed by C. It doesn't have to be this one, any combination they could make. And they could also do tetranucleotides. I don't know the limit. I'm not sure the limit. I imagine it can't be much higher than this at the time. So again, they could use any bases they wanted. It just, for a tetranucleotide, let's say they chose UUAC. Okay, now Gobay and Khorana, what he did was he said, oh, you know what, maybe I can join these together into long chains. For example, take all these and join them together like this. So each dinucleotide one after another. And so now we have a long chain of RNA that could be translated to make a protein, and we know all the codons that are present. So we can see UCU here and CUC. So UCU, CUC, UCU, CUC. Okay, so there's only 2 codons present. And one thing to keep in mind, I'll say this now, I might end up saying it again, is that in the cell, ribosomes don't bind to RNA and start translating randomly. So they usually bind to the 5' end of the RNA, find the start codon, which we'll talk about in a second, and then start translating. When they do start translating, they read 3 at a time. So here's this 3, then they will move ahead, read the next 3, move ahead, read the next 3. In this experiment, ribosomes can bind anywhere, and when they bind and they start reading, they read 3 codons, 3 nucleotides at a time. So, and if you look anywhere in here, CUC, so let's say the ribosome binds here and starts translating. CUC, UCU, CUC, UCU. We still only see these 2 codons. So there are only 2 possible codons present in this RNA molecule. And for trinucleotides, we see something similar. So let's see this one right here. Now, long chain of UUCs, and we can see 1 codon here over and over again if we look at that frame. We'll talk more about reading frames next lecture. So UUC, UUC. Now there's another one here, UCU. And there's a CUU in here too, right? CUU. Okay, so in this long chain of UUCs, we can see 3 possible codons, right? We can see UUC, UCU, and CUU. And down here, we can join these together and get a long chain of UUACs. And with this one, I think we're going to see 4 codons possible. So let's say UUA, CUU, ACU, and UAC. You can look for others, but I've looked at this many times and there's only 4 present. UAC. So we can, for example, we could start here. Look at these 3. UAC, that's present here. UUA, that's present here. CUU, that's present here. So there are only 4 codons in here. So Gobay and Khorana, what he did was he used these with the protein-producing cell lysate, right? And what he found was that these guys right here, when they were used, the radioactive proteins contained were formed when leucine was added and when serine was added. So none of the other amino acids resulted in a radioactive protein. So that means UCU and CUC, each of these must correspond to either leucine or serine. So one other thing before we go on, I will talk about this more later, but the genetic code is unambiguous. Unambiguous. That means one codon can only code for one amino acid. It can't code for two. So that's an important concept as you go through this experiment or learn about this experiment. One codon can only code for one amino acid, or a stop codon, which we will talk about more later. So we don't need to worry about UCU. It'd be impossible that UCU codes for both of these. UCU has to code for one or the other. And then whatever UCU codes for, CUC has to code for the other one. So for these right here, you found phenylalanine was incorporated into a protein, leucine was incorporated into a protein, and serine was incorporated into a protein. Now, how did he know this, right? He used PPCl with radioactive versions of phenylalanine and saw that, okay, phenylalanine was incorporated into a protein, leucine, and serine. None of the other radioactive amino acids, the 20 possible, were incorporated into a radioactive protein. And for these, I won't give the details. I will just say leucine, threonine, and tyrosine were incorporated into a radioactive amino acid or into radioactive proteins. So of these four, there's probably two of them must code for one of the amino acids, and the other two must code for the other amino acids. So now Gobai and Khorana did, I don't know how many he did, let's say hundreds of these types of experiments using different dinucleotides, of different combinations of nucleotides in here. And he was able to work out, or link up all these possibilities with possible amino acids. And then what you can do, or what he was able to do, is just use logic and puzzle building skills, puzzle solving skills with this type of data. And so what we're going to do here, I want you to try, you should be able to match up 4 codons with 4 amino acids from this data alone. We should be able to conclusively determine which the amino acid coded for by 4 codons of all the codons pictured here. Now we can't solve all of them from this data set alone. We would need to do more experiments, but we should be able to solve, link up 4 codons with 4 amino acids. So see if you can stop the video and try to work on that problem for a moment. And then I'll show you how I would go through this problem. I'm a little nervous. This part of the paper is coming out of frame here. I wonder if I could tape this down. And I usually have tape everywhere, but of course now I have no tape. Okay, I'll hold it. I'll hold it down. So, okay, what would I do? Well, first thing I would do is find a marker with a different color, and I would see where we have codons in common between the different chains of RNA. So we have a UCU here and a UCU here. Now what this means is that whatever amino acid we see up here, we're going to also have to see that down here, right? Because the genetic code is unambiguous. UCU here, okay, we're going to need to see whatever amino acid is coded for by leucine. We're going to need to also see it down here. So we've got that one, and I need another color. Okay, so we also have, let me see, I think we have, okay, CUU here and CUU here. Okay, so again, whatever amino acid we see down here, we're also going to have to see that up here. because CUU can code for one amino acid only. So now this is where it gets a little difficult. Let me see if I can remember exactly the best way to solve this. Usually takes me a while to see it. Oh, okay. Now look at the amino acids over here. Leucine is here, and leucine is here. So there are no other amino acids in common between these two, right? So we have one amino acid in common and one codon in common. So CUU must code for leucine. That's the only possibility. Because CUU is present in both, we need to see the same amino acid in both of these being produced when both of these chains are being translated. And okay, leucine is the only one in common. So CUU must code for leucine. Now, when we go back up to here, we see UCU and UCU here. Okay, so we have to have an amino acid that is shared between these two. Oh, and we see serine down here also. Let me think about this for a moment. This is one of the hardest problems. We have some other examples that are in the lecture notes that are easier, so I like to go over the hardest one with you. Okay, so here's the logic here. We have two amino acids here other than leucine. We have two codons that we haven't assigned to amino acids. So each of these two codons need to go with either of these amino acids, phenylalanine or serine. UCU is also found up here. So whatever these code for, whichever one of these two, this amino acid, phenylalanine or serine, whatever UCU codes for, this one, either phenylalanine or serine, also has to be found up here. Only serine is found up here. So UCU must code for serine. Okay, why? Why must UCU code for serine? Because serine is found up here and UCU is also found up here. What about leucine though? Leucine is also found down here. Yes, but there are 3 amino acids and 3 codons. And we've already accounted for leucine here. There's no other way that CUU has to go with leucine because CUU, because of what we found down here. So that leaves us with 2 codons and 2 amino acids. And because UCU is found in 2 locations, we need to put UCU, we can't put it with leucine, we've already accounted for that. We need to put that with serine here and here. If you think this is complicated and you're having trouble following, well, you're correct. It is very complicated. And this is, one of the reasons why Gobay Khorana was awarded a Nobel Prize. And he was working with a much bigger data set. And he was able, essentially, okay, before I go on, let's solve the others. Now this one's going to be easy, right? We only have one other codon here and one other amino acid. So CUC is going to go with leucine. And this one right here, we only have one codon that we haven't assigned anywhere. UUC is going to go with phenylalanine. Okay, so that's how we can use data like this to figure out how, which codons go with which amino acids. And essentially that's what Gobay Khorana did, using massive data sets with lots of different chains of dinucleotides or trinucleotides or tetranucleotides. Giant puzzle, and he solved it, and he was able to solve the genetic code, linking up, confirming previous results that others had suggested that were likely based on other types of experiments. Solved the genetic code and won the Nobel Prize.